Okay, hi. I'm Richard Coppola, and I am director of the MEG Core facility, which is one of the sort of shared neuroimaging instruments in the intramural research program of NIMH and shared also with NINDS. And I'm going to give the summer overview of MEG and describe how it works, give you a little flavor for that, and some examples of how we use it in neuropsychiatric research. So as those of you who are, I see a couple of users in town, I mean, I may be the core director, but the person who really keeps it running and going is Tom Holroyd. And I'll tell you about the rest of the staff a little later on. I'm happy to take questions, just if you shout them out or raise your hand or something. I don't mind being interrupted at all. Please do, particularly if I'm saying something confusing. MEG is just one more of the many possible neuroimaging modalities that we have these days. Most of you are probably familiar with fMRI. You've been hearing things in this course from DTI and spectroscopy. There's NIRS, which is near infrared as a means of looking at blood flow. There's just lots of different ways. And so they're all sort of complementary. I don't think they're really in competition. They have different aspects of how they can resolve the data that we're interested in, because it's sort of time and space that we care about. The temporal resolution of MEG is like that of EEG, that is the electrical activity of the brain. So we can record down to millisecond time resolution, or even faster in the case of things like brainstem evoked potentials. So that part we have covered pretty well compared to the other modalities. The spatial resolution it shows a sort of a wide bar there from about 2 to 4 millimeters, and one could argue about that a lot, depending on really, as in many questions, the answer is it all depends. You can get very exquisite spatial resolution in MEG if you're willing to give up some time resolution. And if you really want to keep that millisecond resolution, then your spatial resolution goes away. It's a little bit like the Heisenberg uncertainty principle. You can't quite have both at once. So it's a bit of a trade-off. And it depends very much on your experimental design. And I'll discuss that a little bit more as we go along. Because it's much more dependent on exactly how you do your experiments and what you're interested in compared to the other modalities. MEG has a bit of a history. EEG, which was started by Lord Adrian back in the 20s with tube and things to measure the electrical activity recorded from the scalp. Well, MEG magnetoencephalography is going to record the magnetic field that is generated by that same electrical activity in neurons. The first machines, the first sensors to be able to do this, to have enough sensitivity, was a single channel SQUID. SQUID stands for superconducting quantum interference device. And the reason you need this very interesting little piece of semiconductor gear is because the fields that we're trying to measure are femtotesla. That's 10 to the minus 15 Tesla. So if you've been down in the NMR Center, you see you've got a 3 Tesla magnet in one corner. Well, we're trying to pick up a signal that's 15 orders of magnitude smaller than that. So we also use liquid helium, just as they do. They use the liquid helium in order to be able to generate the super high fields by having superconducting wires carry a lot of current. We use superconducting, but for the exact sort of opposite reason. Our niobium wire transduces that magnetic flux, that tiny field, into an electrical signal that we can look at. Squids came along, and then with a single channel, and that was sort of, they actually put it over a heart because that's a much bigger electrical signal, and that was sort of the first biomagnetic field that they recorded. But to go from the brain, they really wanted more channels so that you could kind of map that field out. And the first multi-channel one was an 8-channel SQUID, which had a small sensor that could focus on just a little spot. But if you really wanted to map the field, you'd have to move this around. That was quite a nuisance. Then somewhere about 20 years ago, they started building the first sort of whole head multi-channel SQUIDs. And that's sort of what we have, and ours is sort of the latest evolution in that. And I'll describe it a little bit more. This is what our machine looks like. It sits inside a shielded room. It's actually right in the NMR center, not across from the 3 Tesla magnet. It's not the optimum place you would stick an MEG machine. At Tübingen, they have it in a wooden hut out in the middle of the field so that it's far from any other pieces of metal or electrical activity. But we have a 3-layer mu metal shielded room, which helps, and we also have a set of there it is. We have a set of the sensors for the head are in here where the head goes. But up in here, there are 39 additional channels that sample the ambient field and subtract that out. So that's what gives us our ability to operate in this place with a very noise immune. All the signals for stimulation and things have to be projected in from the outside, much like as you do in fMRI, because we're trying to keep all of the electrical noise outside of the room. So what we're looking at is really the electrical activity, direct electrical activity of the neurons. We're a little bit more of a direct measure compared to blood flow. The blood flow changes in response to the metabolic demands that are caused by the electrical activity of the neurons. We're actually recording signals that are directly generated by the electrical activity of the neurons. In the case of EEG, that electrical current that's generated by a neuron firing is volume conducted to the scalp, and you're picked up by scalp electrodes. But if you remember your right-hand rule from high school physics, if you've got a current flowing in a wire or an axon or a neuron, like my thumb, it generates a magnetic field at right angles to it. So that's the magnetic field that we're going to pick up. And so there's a little bit of differences in terms of the current flows that we're actually very sensitive to. We actually pick up all of them, but we're sort of differentially okay, there it is. Differentially sensitive. The EEGs pick up the extracellular currents more because they're volume conducted outside. We're more sensitive to the intracellular current flows. The squid itself has a pickup coil that is made out of the niobium wire, superconducting, and sits inside a vat of liquid helium, so it's superconducting. The squid itself is actually a Josephson junction. It's a piece of sort of transistor-like material that transduces the tiny, tiny signal in here into an electrical signal with a higher impedance that we can record separately. There are different types of what we call flux transformers. The simplest is a single coil, which is a magnetometer, and that's picking up directly the BZ, that is the absolute value of the magnetic field flux that crosses that coil. The other type are what are known as gradiometers, and they actually have 2 coils, and they're counterwound, so that when this field comes through, what it's actually transducing is the delta, the difference between these two, between the field as it goes from spot A to spot B, here from the first coil to the second coil. And that's why it's called a gradiometer. So we're not measuring the absolute value of the magnetic field, we're measuring the gradient of the magnetic field. The reason for doing this is because that gives you another additional piece of noise immunity. And so ours are actually, we have 275 channels of these axial gradiometers. So the gradiometers are wound on a little spindle here. The one coil is down here, and the counterwound coil is here. And then these wires are led up to the squids themselves. This is actually from the CTF. Our machine was, the original company that made our machine was called CTF. This is their 151 channel machine, which was the state of the art at the time when we were interested. We said, well, we're the intramural program. We really want to be on the bleeding edge. So we had them build a 275 channel machine, which is really the same, looks exactly like this, except the coils are a little bit smaller. They're more densely packed. And so it gives us additional set of channels and better signal processing that we can do with the additional channels. This is actually what ours looks like if you're looking in the end. So these are 275 radial axial gradiometers. I say axial because they're organized this way axial to the brain. There's another machine called the Electa, which uses planar gradiometers, which are this way. They have some interesting properties, but you can't space fill. You can only fit so many of them. You only fit 100 of them over the head, and you can't get that many more channels. So I prefer this design, but there's pluses and minuses to each of them. The baseline, that is the distance between the two coils, is about 5 centimeters. And what this does is it puts the focal point of the current that it's going to pick up 5 centimeters below the inside coil. Well, given the superconducting, given the thermos bottle shield and the depth to the cortex, that 5 centimeters is about the distance to the cortical surface. So our maximum focus for our signal is at the cortical surface. We pick up signals from the whole brain, but the maximum spot, if you were to look at just sort of a gain curve, would be at the cortical surface. So this is just a two-dimensional sort of topogram of the 275 channels laid out, just so that we kind of know where they are. The thing that's very important to remember about axial gradiometers is that, see if I can find my cursor again, is that if you pick a particular channel, like F13, the signal that it's picking up is in a circular array around it. The signal right underneath the axial gradiometer, it doesn't really pick up that signal. It picks up a signal from around it. So you can't read this in the same way that you would read an EEG saying, oh, gee, if I see a big signal at one of these leads, the signal's underneath it. It's not at all the case. It has to be processed, and I'll tell you a little bit more about that. The signal itself looks exactly like EEG. I mean, if you people here may be a little not aged enough to remember the days of polygraphs where you had a paper tracing with ink pens scribbling out the EEG trace as it goes by on paper. But this is exactly what it would look like, or on a computer screen. What you're seeing here is an alpha burst. So this is given the time scale. This is about 10 cycles per second. This is sort of the resting rhythm of the brain, known as the alpha rhythm. And you can see the fairly large, somewhat sinusoidal-like looking signals. And this changes with respect to all sorts of things relative to what's going on. This is an eyes closed resting scan, so your alpha becomes maximal in that case. I've only put up a few channels, because if you put all 275 on there, it kind of gets a little busy to pay any attention to. You can do evoked fields with this. This is a, this was a, this is looking at that 275 topogram. Blue is flux going into the brain. Red is flux coming out of the brain. And that's sort of the standard sort of what looks like a dipole, a magnetic or electric dipole, which would be right in between these. These are all 275 channels. This is from at the time of a single spike from in an ictal event in an epilepsy patient. And this is the field map. This field map over here is at this where the red cursor line is, and that's through the spike of the spike and wave from that. So if you want to kind of take all the channels and see what it would look like, you can color code into this kind of topogram. We need to know where this person's head is in order to understand how our signals are coming from the anatomy. So we place a number of coils on the head at the nasion. That's a very sloppy job. I hope none of our users actually do it like that. What I mean is that it should be right on the nasion, which should be right in the middle there, not off to the side like that. And then we also put them on the preauricular points according to a little rubric that we have. We can locate the signals from these coils exactly, and that tells us where the head is relative to the set of sensors. And then we can also image these coils with vitamin E capsules in the MRI and use those as fiducial points to align our MEG reference frame to the structural MRI of that person. So that's what's shown here. So this is that Nasion capsule, except what's done instead of the coil, we've pasted on a vitamin E capsule at that same spot. And then these are the preauricular vitamin E capsules. And so if you locate those and click on them this is actually a homemade program that's part of our CTF software, but AFNI has a whole set of tools for using fiducials like this to align things, and that's how we get back from our spatial reference of our MEG coordinate system and reference it to that person's exact anatomy. So in order to be useful, you want this anatomical brain scan. So you want a good structural. We need it for two reasons. One is because we want to do this alignment. We also need it because although the head is transparent to the magnetic signals, there's a lot of the return currents are in the skull themselves, and that has to be modeled in order to solve our problems for taking the signal that's out here and getting an idea of where the current distributions are inside the brain. So one of the things we want to do is be able to position people similarly in two different times. Although we can realign all of these things and we operate in source space rather than sensor space, it's nice to be able to put the person back in the same spot relative to the MEG. So this is a little program that Tom wrote, and the three boxes are the three fiducial points, and it will store the fiducial locations from the previous scan, and then you can get the person to move and get those back into the same spot. So although we don't have to do this because we can transform spatially the coordinate system, it's better if you have the same coverage. That is, it's better if the person's head is in respect to the coils is in the same spot, then you know you've got the same coverage and you're not going to introduce anything. This is although this looks a little cartoonish, this is absolutely accurate. This is a CAD CAM drawing of our coils. Okay, and this is we won't mention who that is, but that's a surface peel from that person's structural MRI. So this is, although like I said, although this looks cartoonish, this is actually very real relative to the data and the coordinate system. There's a lot of different ways to process this data, and I'm not really going to go into them all. I'll just give you a little flavor and show you some examples, sort of beyond, as they say, it's beyond the scope of this talk. But basically, most of what we're doing is starting with the time series, because what we've got is 275 channels, usually sampled at 600 or 1200 samples per second. So we have this very enormous multidimensional time series. So the start to our processing is always in the time domain. And there's a lot of different things you can do. If you're looking at individual waveforms or spikes, like I showed you, you can do something just at that. Or what is more interesting these days is really to look at the rhythms of the brain at particular oscillations, like the alpha rhythm that I showed you. But we do a lot of processing in a number of different bands. Each band reflects different aspects of functional, either connectivity or functional capability in different regions of the brain. And I'll go a little bit into that. These ones that are listed up here are sort of the classic ones that are left over from many, many years of looking at EEG, the electrical activity of the brain, and where they discovered that theta, alpha, beta, and gamma really reflected different processes. And so by frequency filtering into those bands, you just separate some of those processes, and it helps you to look at different functional aspects. Nowadays, we divide that up a little bit differently. One of the things that we're extremely interested in is high gamma, which is actually above 50 hertz. And there are differential notions about short range versus long range connectivity in the brain that are reflected and also feed forward and feedback that are been suggested by a number of people are done in different frequency by different frequency bands. So we can do evoked fields. That is, if we're going to provide a stimulus that has an event in time, like a tone pip or a light flash, then we can time lock to that and signal average. That's one of the things we can do. But most of what we do is really is more interested in trying to take that data and get a true three-dimensional organization in the brain. And there's many ways of getting from our sensor space out here to the source space in the brain. The simplest one, in a sense, is a dipole fit. So a dipole is sort of a model of a small battery in the brain, which is how you can think of all the neurons that are firing, although it probably takes about 100,000 neurons before you have a signal big enough for us to actually pick up. This is really used when you've got just some focal sources. So in other words, if you're sounding a tone, then you've got a focal initial source in auditory cortex. So locating the source generator for that fits the dipole model very well. So it's best, the dipole fits are best if you're doing sort of primary sensory motor type investigations. However, most of us are really interested in cognitive neuroscience, and these processes are much more diffuse, and also the events are much more diffuse. Although you might flash the stimulus at a fixed point in time, the brain is not always, particularly if it's a more complex task like working memory, it may not be working at one little point in time. So we take our frequency bands and we do something, there are many different types or flavors of how to get back to locations in the brain. The particular one that we primarily focus on is called SAM, which stands for synthetic aperture magnetometry. It's what's known as a beamformer technique. So you're taking all the signals outside the brain and you're forming a beam with them to a specific spot in the brain. And you scan that beam throughout the brain. And that gives you, if you solve for the source at each one of those spots, you then have a full 3D-dimensional picture of the current distributions at the sources in the brains. The other, you'll see in the literature and various other things, a lot of different names for different techniques like this. One is LCMV, which is a linear combination minimum variance, and that's a different type of beamformer. There's also what's called MNE, which is a minimum norm estimate, and that's what's used mostly by the neuromag things. But all three of these, SAM, LCMV, and minimum norm, are if you're thusly inclined, you can actually reduce them all to the exact same mathematical formulas, and it's just a little bit difference in how you are regularizing the matrix underlying this. So they're really very similar, but they make slightly different assumptions. Again, the important thing to remember is that these are estimates, and so these are model estimates of what's going on in the brain. They're not absolute pictures. And so you got to make sure that your particular model estimate is a relevant one for the kind of experiment you're doing. Here's an example of the dipole fit. So what's happened here is that a tone pip has sounded, has been presented binaurally in to both ears. And this is the evoked field that you get if you just take all the channels and signal average to an event marker for, say, 100 of these tone pips. And then this is the pattern, this is the pattern that you would see on that sort of two-dimensional topogram. And what you're seeing are two dipoles. Again, if you remember kind of that right-hand rule I said, if you got a current flow here, the field wraps itself around it like this. So the blue and the red are that field wrapped around. So the current source is actually right here underneath. That is where the current dipole is that is generating that magnetic field. Red is in the brain. The red is actually outside, is flux going out, blue's flux going in to the surface. These are of opposite pictures because of the way the dipole goes. If you now do some mathematics to kind of locate that dipole, a best fit to the outside thing, you get two dipoles like this. And that's the primary auditory cortex, which is the primary signal for being transduced. So, and then this is a similar example. This is now to a median nerve stimulation. So this is a punctile electrical signal to the median nerve. This has there's a number of different pieces to this. This is the N20 component, or actually it's not N, in this case we call it M20 because it's the magnetic field. We don't differentiate between negative and positive because the flux going in and out is not quite the same thing as a negative signal on the brain like the thing is. But if you take keep losing my cursor, if you take the signal right at the red line cursor here. This is just some of the channels here, and you can see that and then you map them out on the brain. This again is a very typical dipole pattern right here, which if this was, we often do right on right. So this was left median nerve stimulation, and that's right somatosensory cortex. So it's exactly where you would expect it to be. So we could do, that's what I meant by if you give up your time resolution, you can really do exquisite localization of where events are in the brain. And just to give you a further example of that, this is a visual stimulation experiment where we're going to flash this little checkerboard, and we're going to flash it in each of the 8 octants of the visual field. So we've asked the subject to stare at a dot, stare at the dot in the center of the screen, and then we're going to take this little checkerboard and flicker it at each of these 8 locations. Okay, the fact that we flicker it at like 7 hertz gives us the ability to get a tremendously valuable signal rate. So we just extract the 7.5 hertz signal from the MEG, and that's got to be due to this flickering checkerboard. So that's how we get really high signal-noise ratio. We've given up our time resolution because now this is just across all of the signals, but we know where we've done the stimulation, and now if we do a dipole fit to that particular signal. And we take the 8 octants, and this is an exploded view of the cortical mantle from the left and right occipital pole. And these are the 8 octant locations. And if you're a visual psychophysiologist, so they will tell you that that's exactly where you would expect the primary visual cortex to be retinally, and in terms of retinotopic mapping onto the brain. So this is what I mean by millimeter resolution. Those are only a couple of millimeters apart, those spots, and we can separate those 8 octants. But again, we've given up time resolution to get that spatial resolution. What we do with the beamformer, because that's a little more interesting, because most of us really care about cognitive processing in the brain, which is a lot more elaborate relative to focal dipoles. So basically what we're going to do is we're going to create a set of weights that enable us to beamform to a very specific location in the brain. And that's sort of meant by the linear coefficient minimum variance beamformer. We're going to take a linear combination of all of the data from out here with an appropriate set of weights that steers the beam to a specific spot in the brain. And that's how we get it's a little bit different than the dipole. One of the ways we can get a better signal-noise ratio is to understand the time window that we want to look at and to understand the frequency band that we want to look at. So what we usually do before we actually do the beamformer, we do what's called a Stockwell transform. And we take all of the data from all of the sensors or some set of them and map them out over time versus frequency. And the color here is the amplitude, your amount of power at that frequency. So this gives us a chance to say, okay, look, we've got a component here that's in the alpha range, and it occurs over this time. So we can set that box, accumulate the cross covariance matrix across all the sensors, which is what we need to compute our weights, and that by honing in on that spot, it gives us a lot of improved signal and noise ratio. So this is again sort of a cartoon. Whoops, the equations didn't come out. All right. Well, you probably really don't mind the equations should have been down here. So you take the MEG data. You do this bandpass filter, you accumulate the covariance matrix based on the Stockwell. We generally do this in 2 different states. So if we're interested in working memory, we could do it where there's a high working memory load, where there's no working memory load. We contrast those, and the map that we will get out is the beamformer distribution, the estimate of source current within the brain. And then, because we've done the alignment of our MEG reference frame with that person's structural MRI, we can take that three-dimensional distribution and map it back onto that person's MRI. And this is sort of what that would look like in a simplistic fashion. I'll show you a little better, some better examples in a moment. But the idea is that we now can take the beamformed source distribution and put that right back on that person's MRI. Can't be a talk without the infamous NBAC. So this is one of the major, sort of cognitive tasks that we've been, particularly in my group. This isn't specific to MEG. This is more specific to a lot of people who do schizophrenia research, do a number of different neuropsychiatric researches, where we're interested in memory processes in the brain, which again now are no longer focal spots. There's a distributed process, but we need to have some idea of what's going on in working memory. We control for it by using what's called the NBAC task. The NBAC task is where we present 4 numbers on a screen. The person has to remember the number and press the button. If it's 0 back, he doesn't have to remember anything. He just presses the button in response to the stimulus he sees. 1 back, he's got to remember that. On the next trial, he has to press the button that he saw 1 trial back. So by doing 0 back, 1 back, 2 back, you can titrate the memory load. And that gives us a way of kind of mapping the working memory component. So that's the sort of standard quick example of that. The thing you've got to do in the MEG is, in addition to recording the MEG, we record all of the event marks so we know when they occurred in the brain. The subject is pressing a button in response. We record all of those signals so we can parse out what pieces of activity we want to look at by having all of the event marks in the brain. I'll give you an example. This is like the first experiment we did 101 back when we first got the machine, which was we did 16 blocks of alternating 0 back and 2 back, sort of the same way you would do an alternating block design in an fMRI. And the reason we did that was because we already had the fMRI data doing exactly that. So we wanted to repeat this in the MEG and see how good our comparison was. So this is, again, looking at the 1 of 4 numbers. These were normal volunteers, all right-handed, and we were going to contrast 2 back with 0 back. And we wanted people who were doing reasonably well on the task, so they did at least 65%. It's actually, 2 back's kind of hard, so 65% is not bad. The way we analyzed the data was with SAM. So we did this. Keep losing the cursor. So we did this. That beam-formed sweep that we're going to go through the brain, we can control the resolution, the voxel resolution that we use for that. So we typically use 7.5 millimeters, or 0.75 centimeters. You can use, it just takes you longer if you compute like 5 millimeters or 10 millimeters. It also depends on how fine a grain spatial analysis you want. This was sort of a good compromise. The time window we used was relative to the block. Or we could now kind of time lock to several different things. We had a whole block that was alternated. The stimuli were being presented, and so there's a stimulus event mark. We could time lock to that. We could also time lock to the subject's response, because they're going to press a button in response to what they're doing. And then we could also do the different frequency bands. So we were going to, in typical NIH fashion, let's do them all. And let's analyze all of these comparisons. So we had 12 subjects, I think, in this. I'll tell you a little bit more. But basically, what we're going to do, because we were able to, there it is. Because we were able, 18, sorry. Because we were able to map each the MEG data, sensor space, source space data back to each person's anatomy. If we align their anatomy using the Telerak atlas, there's different ways you can do that, then we could form a group map by transforming everybody's data into a standard anatomical frame. In this case, it was Telerak. And the color that you're going to see, then you make a color map on top of that Telerac atlas. Blue is desynchronization. That means that the power decreases relative to the task statement, or red is a power increase. We're going to do the group analysis by aligning these, doing the Telerac alignment in fMRI. The SAM image is z-scored to normalize because people have very different ranges. And all of this is done, and then a normalized t-statistic across the threshold to give us a final map. It's fairly standard AFNI type stuff. And you probably get enough of that in other aspects of this course or in AFNI bootcamp if you haven't done that already. The fMRI processing, again, you probably hear a lot about that. This was done on the GE, and this is basically a bold blood oxygenation level dependent signal, all structurally aligned, normalized to MNI space. Actually, we transformed our data from Telerac to MNI. I don't quite remember why we used a different one, but anyway, that's a simple transform. MNI and Telerac they're both normalized spaces. They're just the frame relative to ACPC is slightly different. And again, this was a single subject block design, just like the MEG, except now it's the contrast of 2-back versus 0-back. This analysis was done by SPM, which is a classic way that it's done. And the second level analysis was for the significant data at a corrected p equals 0.001. And so these are the two maps you get out. You actually get a full 3-dimensional structure out that you can sit and look at in AFNI. On the left is the MEG map, and so this was exactly. I guess our original data was 18 in the MEG, but we only had 12. These are the identical same 12 subjects that were picked out of the fMRI data set. And these are thresholded at about the same P level of 0.001. And what you see is a pretty classic working memory network of dorsolateral prefrontal cortex and posterior parietal cortex. It's a little hard to truly adjust your P levels to make these comparable between them. But if you're sitting at your like AFNI and you can set the slider for your threshold, your P level threshold, you can also pick any slice in the brain. You can see how quite exquisite these two maps are the same. If you take the MEG threshold and tighten it a little bit, this resolves into two blobs that are almost identical to the ones that you see in the fMRI. And if you relax the threshold a little bit, we pick up this one over here. So it's just, there's some subtle differences in thresholding and other things, but these are very, so as far as localization goes, these are very, very similar. And that's really what we wanted to see. There's some overarching issues about blood flow differences versus electrical neuronal activity differences and in terms of depth and level. And a lot of people have been studying that, but from this kind of simplistic, I don't mean simplistic, on this kind of mean data that is across a big kind of group, the things come out looking really, really the same. So this is what I meant by if you're sitting in AFNI, you could have, this is actually a single subject in AFNI with those two maps. The top is the blood flow map, and the bottom is the MEG map. This is our typical subject. No, it's not. It's our absolutely best one that we ever saw that aligned on a single subject like this. But you can see the pretty close agreement between the activation for this 2-back versus 0-back contrast. The thing that you can do now that you can't do with or that you can't easily do with the fMRI that we can do is we can extract the time courses from those regions in the MEG. And we can look at how those time courses change relative to the task structure. So we can actually, this is actually from a slightly different one. This is a different memory task. This is the Sternberg memory task, where we now can separate out the encoding, the memory, and the retrieval phases. And so we can look at how which portions of the activated locations have activity depending on which part of the encoding delay, memory delay, and retrieval phases are. So that's something you can't quite do with the fMRI. The other thing you can't do, somebody in the audience may recognize this one, was the kind of question that you want to be able to address with MEG that makes it complementary to the fMRI is to understand when you've got multiple activated regions and you're kind of trying to figure out in a cognitive processing sense whether you've got a top-down structure or a bottom-up structure, you need to be able to understand the time relationships between these activities. It's not, again, not something that you can easily do in fMRI. So this was a task way back a little while ago from Alex Martin's lab and Jess Gilbert, who was the person who actually did the actual heavy lifting to make all this work. And this was, they were looking at the change in signal with repeated stimulation from same objects. So they were contrasting novel objects versus repeated objects, and you get a different signal for the two. And you get signals both in the inferior frontal gyrus and in the fusiform. And the question was, is this top down or bottom up? Well, we could locate I keep losing my cursor. Okay, so we can separate out the novel and repeated signals. We can see the difference in amplitude between the novel and repeated, but we can tell where the time is. We can see that in the inferior frontal gyrus, this is happening at an earlier time than where the amplitude difference is happening in the fusiform. So this suggests that this is a top-down modulation of the process. So it's an example of why you want to use MEG as a complement to your fMRI. Our fMRI here gives us faith in these locations because those are the locations you find from the fMRI, but now we can apply our time capability to sort those out. So those were the kind of very early experiments we did to make us, convince us that this was a useful thing. I'm going to speed up here a little bit. Most of my own work has been in relationship to schizophrenia, so I kind of have a hat as an investigator in what was at that time the clinical brain disorders branch, and we were looking at differences in patients with schizophrenia and their well siblings. And I won't go through the whole thing. I'll kind of cut to the differences here. So what we were doing was bringing in a proband with schizophrenia, their well sibling, and then matched controls. And this is that same N-back task. This is the contrast of 2-back versus 0-back. This is now group data across quite a large number of subjects. And what we see in the normal control is that same pattern that we saw before, bilateral dorsolateral prefrontal cortex and posterior parietal. The blobs here are pretty big because now I think this was about somewhere around, I don't remember the groups, but this was upwards of 60 to 100 subjects or something like that. And so at this threshold, the activated regions are quite big and quite smooth. The patients with schizophrenia fail to activate the LPFC, even though they're still attempting to do this task. And what was interesting, because what we were really looking for was an intermediate phenotype to be able to do a genetic study on this, was that the unaffected siblings of the patients fell somewhere in between. And so this is just, this could be done straightforwardly with fMRI, but we were able to do it with MEG, and then we were able to look at some of the time courses of this data. But it just gives us a way of an example of a kind of, again, early, very basic neuropsychiatric kind of study we did in schizophrenia. This is just the ANOVA for those three differences and showing you where the ANOVA differences are, which was, although it wasn't quite DLPFC, the peak for the ANOVA peak was actually in Brodmann 6, but it's very close by. The other thing that we can do is relate this to genetics. Again, this isn't as specific to MEG, but the reason for the sibling study was to do genetics on these subjects. and one of the early working candidate genes that we had was COMT, which modulates the reuptake and inhibitor, the reuptake mechanisms for dopamine in the brain. And so, again, these are now our group means with different genetic backgrounds. And this is a functional SNP, so the enzymatic activity of the dopamine reuptake is quite different. And the Val-Mets are the heterozygous group. They look most like our normals. This is a bit of an inverted U, that the Val-Vals and the Met-Mets sort of have less frontal activation. We were kind of surprised by this because we thought we would see a straightforward linear curve. Again, not so specific to MEG, but specific to this type of neuropsychiatric research. I'm just going to run through some of the kinds of studies that we've done. Again, this is looking at sodium channel gene versus MEG activity. And again, we find that we've got this is a very similar region that we see, and that follows in terms of the two different allelic variations on the sodium channel. I mentioned gamma power. This is actually now a higher gamma band, not very high gamma, but this is a difference in, this is sort of resting state data. This is not connectivity yet. This is just localization on resting state data. And we have a difference in this sort of slightly higher but not very high gamma band difference, which shows a deficit of gamma power in precuneus in schizophrenic patients. This is now the high gamma that we're starting to look at a lot. So if you actually very carefully map out these higher regions. This is 2-back, 0-back, and resting. And you can see that there's actually out here, this is a much higher frequency. We're actually looking at 65 to 115 hertz, that you get reliable task-related gamma differences out very high. Now, this is the kind of thing that you can't resolve at all with fMRI, because you don't know what, you don't have that component to your data to be able to look at. So here's a very specific time-oriented process that you can't see with other functional methods. Again, early studies, this was some of the very earliest work in ketamine, which is Carlos Arati's group is one of our biggest users. They've been doing MEG along with other neuroimaging studies very early on, from looking at pre and post ketamine. One of the first papers that they had was to really come up. One of the first places where I actually started using the word biomarker, which was that what they showed was that I keep losing the cursor. There it is. So that this is the differential beta power in anterior cingulate. And this is the scan before ketamine administration. Ketamine, as you probably know, administered IV is a very fast-acting, rapid-acting antidepressant. Well, what you're seeing here is the differential beta power in the task from the MEG, and on the y-axis is the change score for the effect of the antidepressant effect. But this is from the baseline scan before the ketamine. So now we've got a biological marker. This is a MEG signal that will predict how big the antidepressant effect will be. So this was really very encouraging to the notion that you could have a potential way of telling who would, either for treatment. There's a number of different reasons why you would care about this. It'll really help you understand the underlying mechanisms, but potentially down the road, it could really help you make decisions about treatment on a very individualized basis, and then to follow treatment in that. We've been able also, this is from some very early work by Brian Cornwell, because people really didn't believe that, okay, you can see these cortical surface things with MEG, but really, can you see deeper structures? And for the most part, people didn't really believe that. And that was mostly due to the fact that the other MEG machines out there were not quite as good as our 275 channel machine in terms of the noise immunity that you need in order to get the signal noise ratio for seeing deeper structures. So he did a very classic, this is abnormal hippocampal functioning using a navigator task. It was sort of a human equivalent of the Morse maze task that he developed, and he showed that the amount of theta power in the hippocampus here correlated with the behavior. In other words, how fast they were able to do this navigation task. So again, now we've got a very clear-cut signal from the hippocampus that's relating to the behavioral ability to follow a spatial map. So there's two things good about this. One is that we've got this behavioral correlation. The other is that it kind of reifies our ability to see a signal from a deeper structure, namely a hippocampus. And then secondarily, I shouldn't say secondarily because the primary interest was that this is different in depressed individuals. So in other words, so we can see differences across neuropsychiatric groups at other than just cortical surface. We can see differences in the amygdala and in hippocampus. And this was just, at that point in time, people were beginning to, this was based on the notion of that early paper from Dr. Zarati's lab, that if you can come up with these kind of signals, you may well be able to do translational neuropharmacology. Like the buzzwords in there. I'm going to kind of skip this part. What I really wanted to get to was this, which was our focus now is really moving on to doing connectivity. Given that we have this time sensitivity, this time ability in the MEG, we can take our time envelope signals and use them to look at correlations between regions. And this is sort of a complex area of analysis because our signals are so much richer than the fMRI ones. The kinds of things we can look at as a surrogate for connectivity between regions is much larger. We can look at power correlation, we can look at phase modulation, all sorts of things. So it's a little bit of a trick to kind of sort this out. And one of the persons who's been really doing a lot of work here has been Alison Nugent, who works with Carlos. And she's been looking at ICA and other ways of dissecting this MEG signal from resting state data to look at differences in connectivity across clinical groups. So I don't really want to, try to dive in. This is obviously a little small to see, but the idea is that you can parcellate out the different sort of networks, components that you see in the resting state, and you can contrast them across two different groups and look at the differences in resting state connectivity. So there's a lot of work to be done to kind of sort this kind of thing out, but this is really the sort of the zeitgeist of what we're doing right now, is to try to develop much better methods for this. The other things that we can do is we can do real-time feedback with MEG, because again, this is an exquisite time signal. We can beamform to a particular spot, pull out a signal. We can do this in real time. It doesn't have to be offline later processing, and we can use that for biofeedback. The other thing that we can do, which I don't have the slide in here, is that we can record during things during different neuromodulation techniques. TMS, transcranial magnetic stimulation, we obviously can't do that in our scanner because this is a huge whopping magnetic signal which would blow our squids totally out of the water. We can do studies before and after TMS, just as we do studies before and after ketamine. But interestingly, TDCS, which is transcranial direct current stimulation, which is a direct current electrodes on there, we can actually do that inside the scanner because this direct current, although it's an electrical signal, it's not moving. So it doesn't create a moving magnetic field. So we can actually record during direct current stimulation. So that's another neuromodulation technique that's of big interest. And so MEG is one of the few places where you can really record in real time relative to that kind of neuromodulation. And I'll just finish up that, again, the thing about MEG is that it's very dynamic. This is looking at gamma band activity during auditory stimulation. This is about 1 second of response, slowed down a little bit because otherwise it would go by too fast, from left and right hemispheres. And you can see there's a nice difference in Broca's-Wernicke's area depending on whether this is an auditory language-related stimulus or not. And so this is the kind of thing that you can really try to attempt to follow real time in an individual subject, what's going on. That's the kind of thing we'd like to develop. This is the proverbial best and only subject that ever really worked like that. But it would be nice to have this available as something where you could really, be interacting with your patient and actually see what was going on in that person's brain at the same time. I mean, that would be the way down the road goal of something you could do with this. There's a whole lot of people who go into this, the MEG core. I'm the director, as I said. Tom is really the person who keeps the machine well cared for and fed and helps with a lot of the data. Fred helps with a lot of the analysis of people. Steve Robinson developed some new techniques. He's actually the original author of the SAM technique. Judy's our lab manager. Sam is our current postbac. The sibling schizophrenia data was from what's now called CTNB, which is Dr. Karen Berman's lab. And the other data was from Mood and Affective Disorders, what was at that time called Mood and Affective Disorder Program, which was Carlos Cerati, Allison Nugent, Brian Kroger, and there's a pile of people. Jess is now with that, which is now with that group. I should update. This is a little old, this slide here. But we do a lot. We have a website. Everything's up there, which just details all of the ways you do this sort of stuff. We have an open scheduling calendar, so it's a very easily accessible thing. You got to learn some things. You got to roll up your sleeve and learn a little Linux. It's not easy, but we're there to help. So, thank you. Questions? Interest? Go to our website. I'm in the, Tom and I are in. We're happy to give tours of the lab itself. It's kind of interesting. It's a little bit different than the fMRI. People get worried they have to take all this thing, all their metal out, and they say, well, the subject does because it'll make noise, but it's not because it's a safety issue. It's purely a noise issue for us. People have to take all the metal off because any piece of metal on your body, and if it moves, it makes a magnetic field signal bigger than the signal from your brain. So you've got to take all your metal off, your body piercings, other things like that. But it's not a safety issue. It's purely a noise issue for us. So there's some subjects we can't run if you've got too much metal in your body for one reason or another. Then it makes too much noise. But otherwise, it's a more benign. People sit up. It's very quiet. You don't have the buzz of the fMRI. But we do need fMRI from people to be able to do our analysis. Okay, thank you.